Assalamu alaikum, everyone. In the previous lecture, we started discussing the different types of stem cells. And based on their origin, stem cells could be embryonic stem cells. We have adult stem cells. Embryonic stem cells, they are taken from embryos. Adult stem cells, usually, they are found in tissues or organs, and they could be extracted from whole organisms. And we have induced pluripotent stem cells, which we will be discussing, and these are cells that could be de-differentiated, or they could be made to become stem cells. We also discussed the different classes of stem cells. So we have classified stem cells based on their potency, based on the number of cells they can make, into totipotent. So they can differentiate totipotent cells. They can basically give rise or differentiate into all cell types of an organism. And then we have pluripotent, which are basically stem cells with high potency but lower than the potency of the totipotent. An example of a totipotent cell is the zygote. Pluripotent cells, they are also found in the embryonic stem cells, and they could be induced. We have also multipotent, oligopotent, and unipotent. These are types of stem cells with, mainly they are adult stem cells with lower potency compared to pluripotent and totipotent cells. All right, so now we're going to focus on the properties of embryonic and adult stem cells. So embryonic stem cells, they are typically obtained from the embryo. So remember, in humans, for example, a sperm fertilizes an egg, and the result is the zygote, and the zygote is an example of a totipotent cell. And then the zygote undergoes or progresses through development and undergoes several rounds of, let's say, of cell division, until, like in humans, after, let's say, after 5 days, around 5 days, approximately 5 days, we have the formation of this blastocyst stage. 5 to 6 days, I think, maybe 5 to 6 days. Now, well, actually, it's 5, if I remember correctly, 5 to 10 days. So this stage contains mainly 2 types of cells. We have a layer. So this is an embryo after 5 to 10 days. And the embryo will contain what we call, so this is, here we have these group of cells. They represent the inner. So this stage is called the blastocyst stage. or it's called the this embryo is called the blastocyst. And we have an outer rim of layers called the trophoblast. And these, the inner cell mass, or the ICM, contains the embryonic stem cells. So all these are embryonic stem cells. Okay. So this is the origin of the embryonic stem cells, and these are pluripotent stem cells. Now, if I remember correctly, this trophoblast give rise to the placenta, al-mashima. Okay, so this trophoblast will eventually form al-mashima, or the placenta. So it will form the placenta, or in Arabic it's called the mashima, okay, which is not part of the embryo. Okay, and we have this cavity, faragh. Okay, this cavity is called the blastocoele. Okay. So this is how embryonic stem cells are obtained from the blastocyst stage. Adult stem cells also can be obtained. They are undifferentiated like embryonic stem cells, and they are found throughout the body. So they are found in different organs, or different, let's say, in different tissues. And typically they are found in tissues and in organs because they are used to replace damaged cells in tissues, for example. And these adult stem cells, typically they are, as I mentioned before, they could be multipotent, oligopotent, or unipotent. An example of adult stem cells is the hematobiotic stem cell, the cell that is responsible for generation of blood cells. Okay, and these hematobiotic cells are found in the bone marrow في نخاع العظم, and in the umbilical cord blood اللي هو الحبل السري. There are some mammary stem cells, intestinal stem cells, let's say endothelial stem cells. There are neural stem cells. So many locations within our bodies, and many tissues, many organs, they might contain these adult stem cells. So as I said before, here this figure shows how embryonic stem cells are obtained from the blastocyst. They could be cultured in the lab. Okay, so we need to provide these stem cells with broth medium, وسط غذائي, that contains nutrients so that these cells can start dividing. Same thing applies to adult stem cells. And then these cells could be forced to differentiate into different cell types like liver cells, nerve cells, blood cells, or any other types of cells. Of course, there has been some concerns or debates regarding the use of embryonic stem cells in research, because you have to understand that embryonic stem cells are taken from embryos, and typically these embryos are the embryos that are not the embryos that are generated during the IVF, the in vitro fertilization procedure, and they are no longer needed. That's why scientists, because of the religious and ethical concerns of using embryos as a source of embryonic stem cells, researchers are now more focused on doing studies or researching or doing experiments with adult stem cells, okay, and embryonic induced pluripotent cells, which we will be discussing later on in this lecture. So why stem cells are being researched or studied? Because we can use these stem cells for treating diseases. There's something called therapeutic cloning. So in therapeutic cloning, stem cells could be isolated. So the idea here is if someone has a genetic disease, let's say, and this genetic disease is caused by, let's say, by a mutant gene. And in this case, we want to find a way of correcting this mutation, or remove this mutation, or provide another copy of the gene that is normal. And this could be done by using, let's say, this type of cloning, therapeutic cloning, which involves creating cloned embryos not for the purpose of reproduction or producing like kids or children. No, for the purpose of producing embryonic stem cells that could be used for treatment. Okay. Now, these stem cells could be, so for example, if there is a person with a genetic disease, or a disease that could be cured by embryonic stem cells, the idea is to take maybe sperm from this individual and use an egg, okay? So the DNA of this individual could be used as a donor, and then we have a zygote and this zygote is basically allowed to divide until the blastocyst stage, and now we have the embryonic stem cells. Of course, the donor, so this is the donor, it could be a sperm or it could be just a nucleus from any differentiated cell. So it's not It's not just only the sperm. Maybe it could be the nucleus of a differentiated cell. And then these embryonic stem cells could be taken and they could be grown in the lab. Okay. And then maybe they might be allowed to differentiate into a cell type that will be used to treat the individual. Okay. So maybe someone has damaged, liver cells or heart cells. So they could, these cells, so here we have embryonic stem cells, and these embryonic stem cells could be allowed to differentiate. Maybe here we could say differentiating cells. Okay. Maybe if this individual has diabetes, okay, سكري, so he or she might need pancreatic cells, خلايا pancreas. Maybe we could, the isolated embryonic stem cells could be allowed to differentiate into, maybe pancreatic cells. Okay, let's say, for example, pancreatic cells, or maybe liver cells, or maybe heart cells. Pancreatic, sorry. Okay, and then these pancreatic cells could be, maybe the donor suffers from diabetes, سكر, and these differentiated pancreatic cells could be injected into the body of the diabetic individual. Okay. Yes, you could. Yes, Yatala. Same thing applies if somebody is blind and needs eye cells, if somebody suffers from heart stroke and jalta, and they have damaged heart cells, they could use heart cells. So, but notice that the ethical issues or lies in the, basically in using embryos, because this is an embryo. So the idea is that here there are some ethical, religious, and moral issues surrounding the use of embryos as a source of embryonic stem cells. Okay. But now, an alternative to the use of embryonic stem cells is to use adult stem cells or induced pluripotent stem cells. Okay. So induced pluripotent stem cells are basically, these are somatic cells that could be treated. Okay. They could be reprogrammed, de-differentiated, so that they are converted into embryonic stem cells. Okay. So typically, differentiated cell comes from, its origin is a stem cell, and stem cell becomes committed, and now it's a progenitor cell, and then a progenitor cell fully differentiate into a skin, for example, a specialized cell. So this is a differentiated or specialized cell. Now this specialized cell, differentiated cell, could be converted back into a stem cell called induced pluripotent stem cell, because now it's a pluripotent. by providing this cell with these 4 transcription factors. Okay. So these 4 transcription factors are cloned into vectors, and there are viruses that will carry these vectors, okay, and infect this bacteria. So now these genes will be expressed inside bacteria. These genes are actually transcription factors that will reprogram the cell and convert it from a differentiated cell into a stem cell. So here we have some sort of de-differentiation, reversal of differentiation. Okay. And then, because these are pluripotent cells, they could be allowed to differentiate into different cell types, like fat cells, maybe liver cells, maybe heart cells, or any other types of cells. Okay. So these induced pluripotent stem cells solves the moral, ethical, and religious issues concerning the use of embryos as a source of embryonic stem cells for therapeutic cloning. Okay, it's called therapeutic cloning here because this procedure is used as a therapy, علاج. Cloning here because we are cloning cells, cloning of cells. So basically, you are taking embryonic stem cells and allow these cells to clone themselves or different or amplify their number, and then finally they are allowed to differentiate. Okay, so instead of taking these embryonic stem cells, which are pluripotent, by the way, and allow them to differentiate into the desired cells, now we can take just only somatic cells. So this is somatic cell, maybe from basically from the donor, the individual affected by the disease, and using these a virus carrying the genes encoding these transcription factors, the somatic cell will be converted into a pluripotent stem cell, and this pluripotent stem cell could be differentiated into the desired cell type. Okay, these are these 4 transcription factors are called the collectively they are called Yamanaka factors. Okay. Yamanaka is the name of the Japanese, the family name. His first name is Shinya Yamanaka. He's the Japanese scientist who discovered these, who actually developed the procedure to generate induced pluripotent stem cells from somatic cells. So basically, these genes could be are cloned into plasmids, maybe, or vectors, and they are inserted into a retrovirus. So it's a virus that can infect human cells. Then the virus carrying these genes will be allowed to infect cells, okay, like this. And then it will introduce these genes into the somatic cell. And once these genes are expressed, they will de-differentiate the cell. And now the cell becomes pluripotent. Okay. Again, these are all transcription factors or genes encoding mainly transcription factors. These induced pluripotent cells are used to treat, let's say, certain types of diseases like diabetes, سكري, Parkinson's disease, Huntington disease, Down syndrome, متلازمة Down, and many other diseases. And currently, scientists have found several ways of allowing these induced pluripotent cells to differentiate into different types of specialized cells. Okay. Also, they are being used, these induced pluripotent cells, they are being used in a type of medicine called regenerative medicine. So regenerative medicine is basically the process of replacing. So here we have replacing human cells, tissues, or organs. Okay? To, of course, restore normal functions. So someone, again, someone has a damaged liver or kidney or heart or some neural cells, regenerative medicine could be used to replace the damaged cells or damaged tissues or damaged whole organs, okay? Using stem cells, of course. I don't know what's in Arabic, it's called regenerative, طب التجديد, maybe. طب التجديد. This is one of the promising branches of medicine that might treat, that holds a promise in treating very difficult diseases like Parkinson's, Huntington, Down syndrome, and many others. Okay. So during this chapter, I just want to compare quickly between embryonic versus against adult stem cells. Typically, embryonic stem cells, they could be totipotent, like the zygote, or maybe pluripotent. Adult stem cells, they are mainly, so here we have adult or embryonic stem cells, and here we have adult stem cells. Okay, let me just answer some questions. ما راح هون الخلايا بتكون متنوعة ولا نوع واحد. Here, these are embryonic stem cells, one kind, but you can allow them to differentiate into different kinds of cells. But here, we want them to differentiate, for example, into pancreatic cells to treat diabetes. Maybe for another person, maybe another person has, let's say, damaged liver, so we allow these embryonic stem cells to differentiate into liver cells, for example. Okay? Yes, Jinan, you can take somatic cells and convert them into pluripotent cells and maybe convert them into pancreatic cells, just like here. Okay? But without taking embryonic stem cells from, let's say, from embryos. Now you take somatic cells like this and convert them into induced pluripotent cells, stem cells, and then you grow them, and now you have induced pluripotent stem cells, and then allow them to differentiate. Okay, so this is another way of doing this. Yes, so diabetes, sometimes what will happen is that we could, these embryonic stem cells, whether they are embryonic or induced pluripotent cells, we can easily correct any gene. So we could use also here something we call gene therapy. So sometimes you just only need cells to treat, for example, if someone had a car accident and his or her liver cells are damaged, so all they need is just liver cells. Or someone has a diabetes and all they need is just pancreatic cells that can produce insulin. But sometimes someone has a genetic disease because of a mutant gene. We can take their we could convert some of their somatic cells into induced pluripotent cells, and then we fix the mutant genes. Okay, this is still under research, and many scientists and companies are trying to, for example, now CRISPR is being used for this purpose to fix genes in the induced embryonic stem cells, and then bring back these genes, these fixed genes into these stem cells, and then allow them to differentiate and bring them back to the body of the affected individual. Okay, so we could carry out gene therapy or gene editing, on these stem cells before allowing them to differentiate, or before putting them back into the body of the diseased individual. No, Tala, currently scientists are just using, mainly focusing on either adult stem cells or induced pluripotent stem cells, because again, there are some religious issues or moral issues or ethical issues of using just embryonic or creating embryonic embryos to take embryonic stem cells from them. Okay. So embryonic stem cells, they are considered totipotent or pluripotent. Adult stem cells, they are mainly, some are, they are pluri or multi. In terms of isolating embryonic stem cells, we could isolate large number of cells from embryos. The number of adult stem cells that could be obtained from the tissues or organs of mature organism are less, so limited numbers. Dana, شيل الفيديو, please. limited number of cells, and they are difficult to isolate. It's not easy to isolate these adult stem cells. Sometimes these embryonic stem cells, because of their high potency, if they are injected back into the human body, they might cause, maybe cause immune rejection. So the immune system might attack these embryonic stem cells if they are injected back into the body of an individual or a person. Okay, it's less likely to have immune rejection, so less likely to cause immune rejection if they are. So remember, for therapy, we have to take these stem cells maybe and inject them back into, sometimes into the body of the individual. Sometimes we inject differentiated cells, or sometimes we put back stem cells, whether they are embryonic stem cells or adult stem cells. So this is, you have to understand that sometimes we put back into the body of the diseased individual differentiated cells, okay, or maybe we put back stem cells, and then these stem cells will differentiate inside the body of this affected individual. Okay. So in terms of sometimes injecting embryonic stem cells into the human body, they may cause cancer. This cancer is called keratoma. So sometimes if embryonic stem cells are injected in the human body, they might start dividing without control, leading to the formation of tumor tissue or cancer. Here, less likely. Less likely means the chances are lower to form tumors or cancers. Tumor tissues. Cancer and tumor, same thing. Okay. And the last thing is that we have some, moral, akhlaqi, ethical, and legal, حقوقي أو قضائي, legal and religious issues. And here, no, we have less moral and legal, let's say, controversy. Controversy means it's just a cancer. It might, yeah, Hadid. Sometimes, if you put embryonic stem cells into the body of a human, they might, because they can divide indefinitely, they might start dividing without control, and they might cause cancer. What this cancer affect? I don't know. It might affect like different organs. Okay, it depends on the location of injection of these embryonic stem cells, maybe in the blood. Okay. So this is just a quick comparison between adults and embryonic stem cells. So and this is the end of this chapter. Chapter 20, the next chapter is about evolution of genomes. Inshallah, in this chapter, we're going to learn about how to study genomes and what do we benefit from studying genomes. And then we're going to also briefly discuss how genomes evolve and what are the mechanisms that will lead to the basically the changes in these genomes. First of all, scientists have been trying to study genomes, not only the genome of the human, but also other organisms. And scientists have been comparing the genomes of different organisms because this is one of the ways of understanding how why organisms are different from each other, but at the same time why they are similar. So studying genomes is an important branch of science. It's a fairly new branch of science because studying genomes was not basically possible, let's say 30 or 40 years ago. Okay. So the development in sequencing techniques, in computer sciences, in computer softwares, computer hardwares made it possible for scientists to study whole genomes. And to study genomes, okay, so genomes can be studied using a fairly new science called genomics. So genomics is the study of all genes within a cell and the interaction between these genes, or the products, the interaction between the products of these genes. And to study genomics, we need to sequence genomes and determine the DNA sequence of genomes and genes. Then we have to rely on another branch of science or another discipline called bioinformatics. So bio is from biology. Informatics, this is related to computer, hardware, and software. So we need softwares, we need computers to study, basically to analyze biological data like genome sequences, protein sequences, gene sequences, and many other biological data. So bioinformatics is basically the application of computational methods to store and analyze biological data. So biological data, this is, we're talking about sequences, whether they are amino acid sequences or proteins, or maybe structural data about the structure of proteins, DNA, and many others, and many other biological molecules, and many other types of data, etc. Okay. So before we discuss how we can study genomics, let's first discuss how genomics as a science was developed. So in 1990, scientists decided to sequence the human genome. And the human genome sequencing began with a project called the Human Genome Project, which took 13 years. So it took 13 years and almost $1 billion to sequence the human genome. And that began basically the era of omics, or the era of disciplines or sciences that are used to study the complete, basically, set of molecules inside the cell. So the genome sequencing basically the Human Genome Project utilized development in computers, computer sciences, and development in the development of new sequencing techniques. So the Human Genome Project was relied on a sequencing technique called dideoxy chain termination method. Okay, and since then, more and more sequencing techniques have been developed. So to sequence the human genome, a scientist from a private company developed an approach called the whole genome shotgun approach. And this approach requires that the genome must be cut into smaller fragments, and these smaller fragments are then cloned into vectors or plasmids, which is not shown here, but these could be vectors or plasmids. Okay, and then these small fragments are sequenced, and then the sequenced fragments, their sequences will be mapped, and they are used to basically to construct the whole genome. So computer softwares will analyze the sequences of the millions of fragments. So here we have fragmentation of the genome, followed fragmentation and cloning. So all these fragments will be cloned into plasmids. Then these fragments, of course, there will be billions of these fragments, of short fragments. Fragments means qita. And eventually, these billions of fragments will be sequenced, okay? And their sequences will be analyzed by computer softwares, okay? So here we have bioinformatics. And the analysis of the billions of fragment sequences will be used to determine the sequence of the whole genome, okay? Inshallah, you will learn about the principles of these sequencing techniques, or the new sequencing techniques, inshallah, in other courses. But this was basically the approach that was used during the Human Genome Project. It was called whole genome shotgun. Whole genome because we start with, so here it's not, so this is not just, it looks like a chromosome, but imagine you have a whole genome, not just one chromosome, whole genome, all chromosomes, okay? And it's called shotgun. Do you know why it's called shotgun? What is a shotgun, by the way? Do you know what's a shotgun? بندقية صيد. صح? No, a gun is a مسدس. Shotgun is a بندقية صيد. Shotgun. So a gun, مسدس, uses bullets, right? رصاصات. But shotgun uses very small particles, like very small bullets. So in a single bullet of shotgun, there will be very small bullets, okay? And it's called shotgun. It's just like you are shooting the chromosome or the genome with a shotgun, and this shotgun will basically fragment this genome into smaller fragments. That's why it's called shotgun. Okay, so just like you are firing a shotgun bullet into the chromosome or the genome, of course, this does not happen. It's just a metaphor, so that the chromosome or the whole genome or the chromosomes will be fragmented into smaller fragments. Okay. Of course, many techniques, many of the new sequencing techniques are still relying on this whole genome shotgun approach. But we have what we call next generation sequencing techniques that do not require cloning. So in the Human Genome Project, the whole genome shotgun approach relied on fragmentation and cloning, but currently the new sequencing techniques do not need to clone these fragments. They could be sequenced directly. So following the Human Genome Project, many sciences or disciplines were developed. One of these is metagenomics. So the development of new sequencing techniques allowed the development of a new science or a new discipline called metagenomics. So in metagenomics, this is basically sequencing the genomes of more than one organisms at the same time for the purpose of determining the identity of these organisms. We have also, for example, something called the microbiome. The microbiome is basically the DNA of the organisms that lives inside our bodies. So you have to understand, humans, for example, are made of humans are made of human cells plus microbiota cells. So microbiota is basically all the other organisms, all the microbes that live inside the human body or on the human body. So we have inside our, let's say, intestine, we have billions of organisms, of bacteria and many other organisms like bacteria, fungi, فطريات, viruses, protozoa, أوليات, living inside our bodies. And their genomes are called the microbiome. And by the way, inside the human body, we have more microbiota cells than we have 10 times 10 times 10 أضعاف the microbiota cells compared to human cells. So basically, you could think of the human or human bodies. They contain two types of genomes. They have the human genome. So our bodies contains the human genome and the microbiome. لينا شيل الفيديو. لينا موسى شيل الفيديو. So the human cells will contain the human genome and the human genes, and the microbiota cells, they will contain the microbiome or the genes of the all microbes. Okay. So now, because of the development of new sequencing techniques that are faster and they are costing less, and because of the development of very powerful softwares, now we can analyze the genomes and study the genomes of many organisms at the same time. And this is what is meant by the genomics. Genomics is the study of genomes of different organisms at the same time. So for example, in this slide, if we have like a soil sample and we want to know what types of organisms are found in this soil sample, an environmental sample, عينة بيئة, or maybe a microbiome sample. So all what we have to do is to do DNA extraction to isolate the DNA from all organisms that are found in the soil sample. Then we use the whole genome sequencing or whole shotgun approach. So here we have like different genomes from different organisms, and I give them different colors. Then we fragment all the genomes into smaller fragments and we sequence the genomes, and then computer softwares will analyze these genomes and they will determine, basically, they will identify these organisms or these species. Species اللي هو النوع الكائن الحي. Okay. So this field of study is called metagenomics, the analysis of more than one genome from an environmental sample or a sample at the same time. So in order to study large genomes, especially if we're talking about studying more than one genome at the same time, we need bioinformatics. So in bioinformatics, we use computer softwares and computer hardwares to be able to study the or to analyze the large data, the large biological data obtained from studying, from determining the sequences of genomes. So the Human Genome Project basically was a successful project because of the development of new sequencing techniques and the development of computer softwares, and because of development of databases that could be used to store the generated genome data and to analyze and study the genome data. So assemble the genome means we determine the genome sequence by analyzing the, so here, let me show you what is meant by assembly. So this step is called assembly, which is basically is done using computer softwares. The computer softwares will use the sequences of the determined for the billions of fragments to assemble the sequence of the genome, okay? Or determine the sequence of the genome. So development and sequencing techniques, development of computer softwares, computer hardwares, databases, in the internet also. All of these new innovations made the Human Genome Project possible. So now all the human genome sequences and the sequences of many genomes from different organisms are all can be accessible freely by anyone with an internet connection. So all these, most of the biological data, most of the genome sequences, the human genome sequence, the sequences of genes, the sequences of genes or genomes of other organisms, they are all stored in databases, okay? So we have a number of famous databases. We have the National Library of Medicine and National Institute of Health database, okay, called NCBI. We have the EMBL databases. This is basically a European database. We have a database of created by the Japanese and a database created by Chinese. All these databases, okay, all of these they store, okay, and they store biological data, genome sequences, gene sequences, and other protein sequences, protein structures. But also, they contain software for biological data analysis. Okay, so not only they store the biological data for us, so we can access this data anytime we want, but also they provide softwares for us to be able to analyze genome sequences, protein sequences, gene sequences from different organisms or other types of biological data. So for example, we have a database within the NCBI databases. So there are different databases. We have databases for genomes, we have databases for genes, we have databases for proteins, we have databases for protein structures, we have databases for all sorts of biological data. One database called GenBank. So from its name, it's a DNA database of DNA sequencing. And within this database, also, there is a software called BLAST. So BLAST, this is a software that allows us to search for a specific DNA sequence, to predict a protein sequence from a DNA sequence, okay? And maybe to compare DNA sequences from different organisms. Also, there is a software that allows us to compare the protein and DNA sequences from different organisms. So this is a figure that shows a software comparing the DNA or the protein sequences for a protein called DW40. So this WD40 is a protein found in most eukaryotes. And as you can see here, we have a comparison between the same protein from different organisms, from cow, from a weed, ashab, from corn, dhurra, from humans, from nematode. This is a type of worm, dude, from yeast, khamira. And as you can see, these highlighted amino acids are common in all sequences, which means that these are the amino acids that are most important for the function of this protein. This is another software that shows the structure of this protein, for example. Okay. So I will stop here today. Inshallah, we're going to continue tomorrow.